method, inheritance and variation. Today we discuss structure of chromosomes. In that chromosomes which are discovered by Walter Fleming. In 1882. Walter Fleming discovered chromosomes in 1882 in the cell of larva of salamander. In the cell of larva of salamander, Walter Fleming discovered the chromosome structure. And that chromosome structure that previous lecture we saw the chromosomes which are visible in the metaphasic chromosomes. Metaphase. Metaphase is one of the cell division stage, that is the prophase, metaphase, anaphase, telophase, and in that when cell divide, the that is the chromosomes, that is the nucleus, and these chromosomes are present in the form of chromatin network. The DNA present in the, that is the actual in a single cell DNA, the unbonded DNA in a single cell, the DNA is 2.2 meter. That means these 2.2 meters DNA is how it is possible which situated in a single cell. That means these uncoiling DNA or these single DNA, that is 2.2 meter, and which is and it is situated in a single cell. This is the magic of biology. This is the magic of genetic, how it is possible that this 2.2 meter DNA which is situated in a single cell, very very microscopic single cell, because of the histone protein. The histone protein, which is one of the type of the protein, which is the globular protein here, and which is the octamer. This DNA, we know that is the DNA which is the double stranded helical structure here. This DNA which is coiling or bind to the histone protein, then again it is bind to the globular histone protein, which is the octamer histone protein, and then this is coiling and coiling and coiling and coiling and coiling, which is situated in a chromosome here. This is the coiling of the chromatin network. This is the condensed form of the chromatin material. That condensed material. What is the condensation? In cell division we know that the prophase there is much more condensation of chromatin material. Then metaphasic stage there is again the condensation and the chromosomes become very short and thick. That is the which is this these chromosomes are visible in the microscope. That is the metaphasic chromosome, and we know that is the condensation. Then what is the condensation? Condensation is nothing but there is a coiling of chromatin material, or this chromatin material there is a DNA also bind, or DNA also coiling, which is histone protein, and this situated, that is the coiling of the chromatin material, coiling and coiling and coiling and coiling here, and that is the condensation. घनीभवन क्रोमोसोमचं काय होतंय? कॉइलिंग होऊन अशा प्रकारचं स्ट्रक्चर होते, and that structure is visible in the microscope, and this structure, that is the metaphasic structure. In the metaphase, that is the cell division phase. One of the metaphase is the cell division phase, and which is visible in the chromosomes. Then we see, now we see this second is also chromosomes. But this chromosomes is the duplication of the chromosomes here. This chromosomes is the duplication of chromosomes, and this happens in cell division. In cell division there is a duplication of chromosomes. This is also chromosomes and this is also chromosomes here. These two are the chromosomes here. This is the chromatids. That is the two arms of this is first arm, this is single arm, and this centers. That center is called centromere, and this is also centromere. And because of duplication, these chromatids are converted into two chromatids here. That chromatids is called as sister chromatids. Sister chromatids. Now this we see there is no confusion about this is the chromosomes or this is the chromosomes or this is chromosomes here. These two are the chromosomes, but here this is the duplication of chromosomes and it happens only in the cell division. We see the structure of the two diagram here. The two diagrams are the chromosomal diagram here. That first, that is the A diagram, show the part of the chromosomes here. This chromosomes, that is chromosomes, the condensation of chromosome, that is the metaphasic chromosomes here. The center part, which is the spherical part, which is called a centromere. This is the centromere. This centromere, then this part, which is called chromatid, or these two parts, which is the sister chromatid. Sister chromatid. And in between these chromatids, the DNA are situated, which is extend over the length of the chromatids. DNA is a double helical, and these DNA which is extend, which is the length of the chromatids, and this is called chromonemata. What is chromonemata? We first see that this first diagram, which is the A, which is the chromosome diagram, which show the part of the chromosomes, and this second diagram, that is the B, which show the construction or the constriction of the chromosomes. First we see this diagram, this is the centromere, which is situated in between the chromatids. Here the chromatids, which is upper side and downside here, and middle side there is a centromere. Then this centromere, that is centromere, are also called here the primary construction. Here also this is the centromere. This is the chromosomes. This is the centromere. This centromere also called the primary constriction. The primary constriction, pratham akunchan, that is the constriction. Here that side the chromosomes become very short. That is called a constriction here. This is the primary constriction, that is called the centromere, and the primary constriction consists of discus shaped plate, that is called a kinetochore. Here the two discus shaped plates are present either side of the centromere here. These are two discus shaped plate, that is called kinetochore. This kinetochore, when present or when situated, this kinetochore, when produced this kinetochore in the cell division, when cell divide, at that time the kinetochore which are formed. And why kinetochore? Because of called the kinetochore. Then this is the chromonemata. This is the chromonemata. Chromonemata, which is nothing but the DNA. Chromonemata, which is nothing but the DNA, at which is it is means this is the coiling DNA, or this is the coiling DNA. This is the chromonemata, which is nothing but it is a DNA, or which is the coiling DNA. And why it is called chromo? See, the chromosomes, the chromo means color. Chromo means color. Here the chromo means color and nemata means thread. Chromo means color and nemata means thread. Color. And here chromosomes means color. Chromo means color and somes means body. Color body chromosomes means when cells are stained and that chromosomes appear the stains, that's why it is called color bodies. And when chromosomes are stained, this part of DNA which appear the color, that's why this part of DNA then called a chromonemata. It is actual DNA. It is actual DNA. हा खरं तर DNA आहे, but when stained it appear color, that's why it is called chromonemata. It is nothing but DNA. Then this structure, that is the primary constriction here, the primary constriction which is also called centromere, and centromere which is also called here the primary constriction. Then the secondary constriction here. This yellow part which is the secondary construction here, and this is also the secondary construction. This secondary construction, this is the first secondary construction, this is the secondary construction. This secondary construction also called nucleolar organizer, which is why it is called a nucleolar organizer. Secondary construction also called nucleolar organizer. Why it is called nucleolar organizer? Because they are responsible for the formation of the nucleolus here. This construction which formation of the nucleolus here. The nucleolus. This is the primary construction, this is the secondary construction, but the secondary construction is also called the nucleolar organizer. Why it is called a nucleolar organizer? Because it responsible or it form or the formation of the nucleolus here. हे secondary construction काय करते हैं? The nucleolus तयार करते हैं. That's why it is called the nucleolar organizer. Beyond the nucleolar organizer or beyond the secondary construction, the spherical structure, that structure is called the satellite. Beyond the nucleolar organizer or beyond the secondary construction, a short and spherical structure is called satellite, and it is also called sat bodies. Sat bodies. Sat bodies, these sat bodies which is present the preparative DNA. These sat bodies present the preparative DNA and it is used in a DNA fingerprinting. These sat bodies, the preparative DNA present and which is used in DNA fingerprinting. This sat body. Then the telomere here. The tip of the chromosome is called telomere. This is also telomere. This is also telomere. That is the tip side or the end side of the chromosomes is the telomere, and it is the unique property that prevents the end of the chromosomes from sticking together. Or this telomere other. क्रोमोसोम्स हे सेलच्या न्यूक्लियोलस मध्ये ज्यावेळेस तयार होतात त्या टाइमला एकमेकांपासून एका क्रोमोसोम पासून दुसऱ्या क्रोमोसोमला चिकटण्यापासून वाचवणारा that telomere is one of the component here. That telomere which prevent the chromosomes from sticking together. So telomere which prevent the attachment of two or more chromosomes to each other. That is the one of the function of the telomere. Telomere have the repeated base sequence. Telomere which is nothing but the repeated base sequence of T, T, A, G, 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 which is a repeated base sequence. And what time it is repeated? That it repeated 2500 times repeated. That is the telomere. Telomere which prevent the sticking from the chromosomes with each other, and which is the base sequence, it is repeated base sequence, that is the T, T, A, G, 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 and 2500 times repeated. And one of the thing about these chromosomes here, telomeres here, that telomeres have, which is help to calculating age also. Telomere helps the calculating age. That length of telomere, length of telomere which is decreasing, the person age is increasing, or the aged person, when the length of telomere is very short, then the person age or the person is aged person. Aged person here म्हणजे टेलोमेअरची लेंथ जर ॲक्युरेट लेंथ असेल, भरपूर लेंथ असेल, तर त्या पर्सनचं वय हे जास्त असतं. आणि जर लेंथ हळूहळू कमी व्हायला लागली किंवा लेंथ कमी असेल, दॅट इज द डिक्रीजिंग लेंथ, तो पर्सनचं वय कसं जास्त झालेलं असतं. that depending on the calculating age of the telomere also, then which is chromonema, we see here the chromonemata or the chromonema, then here the centromere also, centromere, this part, swelling part, this all parts here, the centromere part, chromomere part here, by mistake I say the centromere here it is the chromomere. And chromomere is nothing but it is the swelling part. And which part? That here, that structure on the DNA, here the swelling part which is present, this is also the chromomere. And chromomere is nothing but the histone protein. When the DNA is binding, and that binding structure, that structure, the histone protein is looking like that. It is nothing but the histone protein, that is the chromomere. When DNA bind, it's looking like this. That part is look like a swelling, and this is nothing but the chromomere. Chromonema is nothing but the DNA, and chromomere is nothing but that is the histone protein part, and it is the swelling part here. This is all are the chromomere. Which is the surface, which is look like the chromosome surface, which is the soling, that is the फुगल्यासारखं दिसते. Chromosome surface look like a soling, and that soling part which is called the chromomere. This is the total parts of the structure of the chromosome here.